During this presentation, I am going to discuss how oncovin binds to tubulin and leads to prevention of cell division, and also how tubulin is used in cell division. First of all, I will go over the CHOP method, C-H-O-P. It is a chemotherapy combination. It involves 4 different medications: cyclophosphamide, hydroxydoxorubicin, oncovin, and prednisolone. It is a treatment for non-Hodgkin lymphoma. These drugs destroy quickly dividing cells like cancer cells. Each medication within this method works on cancer cells in different ways. Tubulin. Tubulin is the protein that polymerizes into long chains that form microtubules. Microtubules serve as a skeletal system for living cells. Microtubules can shift through different formations to allow mitosis. This is made possible by the flexibility of tubulin. Tubulin is a protein that makes cell division and the movement of materials in cells possible. The half-life of tubulin is only about a day, whereas a microtubule may only exist for about 10 minutes. Microtubules. These give shape to cells and cellular membranes. They allow cell movement, such as in the contraction of muscle cells, and much more. They allow the transportation of specific organelles within the cell. They allow mitosis and meiosis. They allow to separate replicated chromosomes to opposite ends of a cell during division. Here's a picture of a microtubule. You'll see that there's 2 different tubulins that kind of combine to form the microtubule. The tubulins are the proteins that form it. With cell division, this is important to reproduce life, makes new cells out of old cells. Microtubules contribute to the formation of the mitotic spindle. The mitotic spindle separates replicated chromosomes. Oncovin. Oncovin inhibits microtubule dynamics. It causes mitotic arrest and cell death. It is a microtubule destabilizing agent. Oncovin also stimulates mitotic spindle destruction and microtubule depolymerization. It can also block mitotic progression. It can bind to both soluble and microtubule-associated tubulin. This can decrease the rate of microtubule dynamics, but it also increases the duration of attenuated state of microtubules. Oncovin also makes sure that the mitotic spindle is not properly assembled. It inhibits chromosomes from progressing to the spindle equator. The progression from the metaphase to anaphase is blocked within the cell cycle. Cells undergo unequal cell division. They may not divide at all. They may undergo apoptosis, or cell death may occur. Here is just a diagram I found that shows kind of what happens during the phases. You go from interphase in mitosis to metaphase to anaphase, but here it shows how it's blocked by tubulin binding agents, causes the mitotic arrest and cell death. The oncovin is the name can also be vincristine. Cancerous tumors are characterized by cell division. Cancer cells lose the ability of contact inhibition. Plant alkaloids are cell cycle specific medications, which is what oncovin is. These attack cells during various phases of division. They also inhibit the microtubule structures within the cell, which results in the cell death. Here is the structure of oncovin that I found. And I also found another little diagram that talks about the alkaloids, vincristine and vinblastine, which is another type of medication that is used for cancer. These drugs block the formation of mitotic spindle by preventing the assembly of tubulin dimers into microtubules. They act primarily on the M phase of the cancer cell cycle. Resistance is due to increased efflux of drugs from tumor cells, so it shows how everything's kind of blocked and then prevents anaphase from even happening, and the dissolution of the mitotic spindle leads to cell death, so they don't even allow it to progress on to the next step in the cell cycle. And these are some of my references. Thank you.